are going to start talking about the nervous system and pretty much talk about the various different parts of the nervous system from now until the next exam. So today, we're going to just kind of do a review a little bit about the different parts of our nervous system. So you remember we have a central nervous system, which is made up of what?
The head and the spinal.
The brain. The brain and the spinal cord. Exactly. This is the central nervous system. And then that central nervous system is going to talk to, interact with the peripheral nervous system. Now, information has to go to and from the central nervous system and interact with the peripheral nervous system, which is basically everything else that isn't the brain or spinal cord. That's what our peripheral nervous system is. So if the information is coming from the body and going to the brain, we say this is afferent information. If the information is coming from the brain and spinal cord and going to the body, this is then efferent information. Okay, so let's take the peripheral nervous system and break it down into its separate parts because it's a big nervous system, okay? And basically it has, the peripheral nervous system has two major components. One portion is what we would call the sensory nervous system or the sensory division of the peripheral nervous system. Then the second major component would be the motor division of the peripheral nervous system. Now the sensory division is all the different sensations of our body. Everything that we feel and taste and smell and touch, and pain that you might have, and light, and muscles are sore, and all those different sensations, whether they're external sensations or internal sensations, those are part of the sensory division. And that information is going to go to the central nervous system. So this information is always going to be afferent. Now, the brain and the spinal cord both are for integration. Now, integration means basically thinking. A lot of people assume that the spinal cord is only kind of like a superhighway for information, neurological information to go to and from the brain. But in actuality, in many different instances, the spinal cord has the ability to also think, just like the brain does. So the spinal cord can interpret this sensory information and figure out what to do about it. And whether it's the spinal cord or the brain, once it figures out what to do about the sensory information, it's going to send information back to the body via the motor neurons, and then we're going to do something about it. So any information for the motor division is going to be efferent information. So this is going to come from the brain, from the spinal cord, go out to the body, and have the body respond. Now, there's two basic divisions of response. One is a voluntary response. One is an involuntary response. So we're going to break the motor division into two parts. So one part is the voluntary response, which is the somatic nervous system. And this is going to be voluntary, which then the only thing that we use for voluntary response are going to be skeletal muscles. The involuntary response then is going to be what we would call the autonomic nervous system. Now, the autonomic nervous system is more than just skeletal muscle, okay? We're going to have information for, let's say, smooth muscle and cardiac muscle and glands. These are going to be the biggest responders to the autonomic nervous system. So both of these, again, are efferent. Information is coming from the brain via the motor division going to skeletal muscle by motor neurons or going to the autonomic nervous system by motor neurons. And we're going to have either a voluntary or an involuntary response. Now, this involuntary response has a lot of different parts to it also. And there's a lot of different ways for our body to respond. And so we have two major divisions to our autonomic nervous system. The first major division is called the sympathetic nervous system. The second major division is the parasympathetic nervous system. Now, remember, the autonomic or the involuntary is all about smooth muscles contracting, cardiac muscle contracting, and glands contracting for the most part, okay? So this would be things like allowing our blood pressure to go up and respiration to go up and perspiration or salivation to change rate. And so the sympathetic nervous system affects all these, but they affect them in opposite ways. So the sympathetic nervous system typically, for the most part, makes things contract or secrete. So if we talk about the sympathetic nervous system, usually things go up, they go faster. So sympathetic nervous system makes heart rate go up, respiration go up, perspiration go up. All kinds of things are going to rise, blood pressure goes up. This is all part of the sympathetic nervous system. Parasympathetic, on the other hand, usually makes things calm down or shut down. So heart rate will decrease, blood pressure will decrease, respiration will decrease. This is typically what's going on between sympathetic versus parasympathetic. Now, a lot of times you'll hear people say sympathetic is fight or flight. Better known to us nowadays as stressed out. Okay? Whereas parasympathetic is much more like resting and digesting. So remember, heart rate has to go down, respiration goes down so that you can rest. But then it also helps to speed up digestion. That's the only thing it really speeds up is the digestive tract with the parasympathetic nervous system. I'm going to put that on silent. Thank you. Any questions about this? Now, we are going to go into sympathetic and parasympathetic in a lot of detail because one of the other things that we find is that they also control our immune system and has a lot to do with our health. And so we want to talk about how this affects our body health-wise because it has a lot of implications to people who have diseases. And so really important for you to understand what the autonomic nervous system is doing to your patients, whether it's helping them or not helping them. All right, so the next thing I want to talk about is just kind of go over a little bit the difference between a neuron and a nerve. Just sort of refresh your memory. So a neuron is a single cell. It's just like the cells we've been talking about. Neurons have a phospholipid bilayer with integral membranes. They have a nucleus. They have all kinds of different organelles, Golgi apparatus, endoplasmic reticulum. everything we've been discussing, the only real difference is they look kind of funky. They're not your typical looking cell. And we're going to talk about how this change in shape also allows these neurons to send information through the body from one place to the other. In comparison to this single cell, though, we also have something called a nerve. And a nerve is just basically a group of neurons. And it could be hundreds or even thousands of neurons all grouped together in one place. Now, if you remember from your anatomy class, many neurons have, not all, but they have this outer kind of fatty layer around it called the myelin sheath. So this is supposed to represent a single neuron right here, like that picture that we just looked at. This is a single neuron, and it has the myelin around it. Now, we're going to talk a little bit more about myelin in a few minutes, but basically myelin is insulation. We're insulating this neuron because this neuron is going to conduct electricity. So, like this cord that I have plugged into an electrical socket right here, and then I have it plugged into my computer, although I just noticed it's not on. Now, there we go. If I didn't have this insulation around it, what would happen if I grabbed a hold of this cord? Shock. How?
There's no covering outside.
There's no outside covering. Where does the electricity go? Into me. What we want is we want it to go from point A to point B. We want it to go from the socket to my computer to keep it charged up. But if I have no insulation on it, then it arcs out. When I touch it, it goes into me. And we call it shorting out, okay? And we call it out, all right? So we don't want that to happen either with our neurons. And we have these insulation sheaths, these myelin sheaths, wrapped around the neurons so that they don't short out. Now we also have a second layer of, a second layer of insulation here. This is called the Endo Neuron. This goes around this single individual neuron. Now inside a nerve, these neurons are bundled together. They're grouped together, and you can see that there's 4 major bundles inside this nerve. And to hold these bundles together is the perineurium. And so you have this connective tissue layer holding our bundles of neuron together. By the way, a bundle of anything in biology is what we would call a fascicle. So if you like bundle your hair in a ponytail, we'd call it a fascicle of hair. Anything that's bundled together. So these neurons are a fascicle surrounded by the perineurium. And then you have multiple fascicles inside this nerve, and it's surrounded by this external connective tissue layer, which is the epineurium. Now, probably, I'm going to guess, when you were dissecting your cats in anatomy, you saw nerves, those white stringy things that went through the cat. Well, what you were looking at was just the epineurium. If you took scissors and cut that nerve in half and tried to look inside, it would just look solid white because you can't see these individual neurons because they're little cells, okay? You can't see them with the naked eye. Very, very small. Inside, we also have some blood vessels that help to feed these neurons all the time. But this is a way to keep neurons together, and each individual neuron is usually in a nerve the same kind. So typically all these neurons would be sensory neurons, or all these neurons would be motor neurons. And so you'll hear people refer to them as sensory nerves or motor nerves. Sometimes there are some of these nerves that have a little bit of a mixture, a little sensory, a little motor inside, and so you'll hear them called a mixed nerve. So we have sensory nerves, we have motor nerves, we have mixed nerves, depending on what kind of neuron is on the inside of this nerve. Now let's just talk for a little bit. We'll talk about motor neurons, but I wanted to give you some examples of sensory neurons because we actually have more different types of sensory neurons than motor. Motor are stimulating muscles. They're for skeletal muscle, cardiac muscle, smooth muscle. But sensory neurons, they basically do everything else. So you don't have to memorize these. We will go through these as we go along. I just wanted to give you a list just to look at right now. Sensory neurons, though, okay, here's a bit of a problem, can also be referred to as sensory receptors. Okay, now remember, we've been talking about receptors as that thing on the outside of the cell that something binds to, and we stimulate the cell, get gates to open, that type of a thing. Now we're going to be talking about receptors as neurons. And here's the weird thing. Neurons, being that they're a nerve, also have these receptors on them. So it gets a little confusing, and I wish somebody had brought up a, named them a different name, but oh well. So when we're talking about sensory neurons, I'll try to be as accurate as I can if I'm using the word receptor, so you'll know it's actually the neuron. So this particular sensory neuron they call a mechanoreceptor. And mechanoreceptors can do a few different things. For instance, they can help you to feel when your muscles are stretching, or they help with equilibrium in your ear so you don't fall over. They help with touch on the outside of your skin, like pain and cold and heat, that type of a thing. Chemoreceptors, these are receptors we're going to talk a lot about tomorrow, and they help us to determine sense of taste and sense of smell. Okay? Osmoreceptors, we'll talk about these next week, and they are all about monitoring blood pressure, or what we'd also call osmotic pressure. Photoreceptors, these are receptors in our eye that allow us to see light. Now, let me just say, every different type of neuron in our body has one job and one job only. So for instance, the photoneurons, the photoreceptors in our eye, help us to see light, but they certainly don't allow us to smell anything. Okay? And the chemoreceptors in our nose help us to smell things, but they don't feel pain.
So in every neuron there's one type of information.
Every neuron has one job, it perceives one kind of information, and that's it. Okay, so you need to know they don't do multiple types of jobs in the body. I thought this was really interesting, so I put this in here. We don't have this, but snakes have infrared photoreceptors. That means they can see you in the dark. So be really careful in the high desert, because you might not be able to see them, but they can certainly see you. What's the nasty snake that we have up here? The Mojave green. It is the most venomous snake in North America. You want to stay way, way, way away from that puppy. And by the way, they're very aggressive snakes. So the Mojave green has been known to chase people. Regular rattlesnakes, you can kind of stand away from them and look at them and be a distance, and they don't care. Mojave greens are like, don't look at me. Okay, they're like the bad boys of the desert. Okay, don't even look at me. Just get away from me. And if you keep looking at me, I'm going to chase after you. And they do. They are very, very aggressive. Mojave greens have two different types of venom, which makes them worse than other rattlesnakes. The first type of venom paralyzes you, and the second type of venom is like Ebola. It actually just starts to dissolve your internal organs. And so you get bit by Mojave green, you need to call somebody to say, get me a helicopter now. Because the other weird thing is we don't have any anti-venom for rattlesnakes in the high desert. You have to actually go to Loma Linda to be given rattlesnake anti-venom, because they're the only ones around that have it, which I don't understand. Every once in a while, if you listen to the country radio station, you might hear Loma Linda making announcements on the radio to ask people to go out and start catching rattlesnakes again, because it's time. They've run out of the anti-venom. And there's a bunch of crazies up here who go around, they collect rattlesnakes all over the desert, bring them down to Loma Linda, and then they milk those snakes, and they make their anti-venom from it. So unless you know what you're doing, do not listen to that radio station. Do not go collecting rattlesnakes. It's just not a good idea. By the way, Mojave greens are green. That's why they call them that. kind of a lime green color. They fit really nicely under the, desert green type of bushes, and they're hard to see. So you just need to be real careful. Thermoreceptors. These are receptors that help us to tell differences in temperature. And this is one of the things we're going to do in lab today. We're going to figure out the density of your different thermoreceptors. So we're going to try to see if you have more hot or more cold thermoreceptors. Because the more of one type of receptor you have, the more intensely you feel that temperature. So for instance, just genetically, some people may have more cold receptors than hot receptors. And usually if you have a lot of cold receptors, you feel cold intensely, you don't like to live in cold places. If you have a lot of heat receptors, you feel it more intensely, you can't stand the high desert in the summer. Okay, you probably are dreaming about going to some place there's snow, because you don't feel the cold as intensely as someone else. So we're going to do a little test in lab today to see which type of receptor you have the most of. Phonoreceptors, we'll talk about these. They're the receptors in our ear. They help us to pick up sound waves. By the way, we don't have these, but I thought this was cool too. Electroreceptors. Electric eels have this so that they can find their girlfriend in the ocean. They can detect electrical currents. So another electric eel's giving off electricity, they can detect that. Sharks have something very similar to that. They can detect changes in wave patterns in the ocean up to a mile away. So when you're kicking and bobbing around and everything, you can know that any shark in the area knows that you're there. That's why I don't go out there. My brother he's crazy. He's a big time surfer. He's been on the surfing circuit and everything. And he told me he was surfing not too long ago off the coast of South Africa, which is a huge place for great white sharks. And he said he was just out there, waiting for the next really big wave, and he felt something hit his foot. And he thought it was a seal, didn't think anything of it, and then a few minutes later, he felt the thing come back the other way and hit his other foot. And of course, being my dumb brother, he didn't think anything of that either, didn't even look down, so he waited for the thing to come back a third time and hit his foot again. And then he decided to look down, and he said it must have been at least 15 or 20 feet long. And I'm like, okay, what did you do? And he goes, well, I picked my feet up out of the water because, they're looking at you from underneath and they're thinking you're a seal because you've got, all 4 arms and legs on this board. They don't know any better. And he said he just was praying for that wave to come really fast so he could get the heck out of there. So he did. He caught a wave. He got out. But he's screaming to the other guys. And my brother's a huge practical joker, so they thought he was just joking. And then I guess that shark must have come up and they saw the fin and, everybody else was out of there too. And I go, okay, so what did you do after that? He said, well, we waited for about 15 minutes and went back in. Oh my gosh. Okie doke. It must be because when he was born, somebody dropped him on his head or something. I don't know. But didn't faze him. He just got right back in 15 minutes later. Anyway, the last type of receptor is a nociceptor, and I really want to tell you about this one because this is a pain receptor. We have more nociceptors than any type of sensory neuron in the body. Our body wants to make sure we feel pain. Why is that? Why? Say it again? To keep us alive. Keep us alive. What does that mean?
If it hurts, you're not going to do anything?
Helps to protect us, prevents damage, right? Okay? So there is a genetic defect that some children are born without any nociceptors. And they cannot feel any pain at all. So imagine, okay, let's say you're walking around the corner of the desk and you walk too close and you hit your leg really hard on the corner. You know next time to walk further around. Okay, your brain makes that conscious adjustment, okay, unless you're just a klutzy dummy, all right? And you make that kind of adjustment. But these little kids don't know any better. They hurt themselves, they hit themselves, they tear skin, muscle, whatever. They don't feel any pain. The brain doesn't know better to make that adjustment. I had some family friends that they had a little girl who was born with this very rare disease. And at 2 years of age, she had some brothers who were like 3 and 4. They weren't much older than her. And they just thought it was really funny that she was running into the garage door. And so they were egging her on, and she just kept backing up and running into the garage door over and over and over again until she killed herself. She didn't know. Her brothers thought it was funny, she thought it was funny, she just kept doing it. And we also see this problem, for instance, in people who have leprosy. One of the problems with this disease is it also kills off the nociceptors. So these people become very deformed because they don't realize that they're hurting themselves. So, you know, they're using a knife, not paying attention, they may end up cutting off a finger and they don't even feel the pain. They might feel a little something, but that's not enough for them to pay attention to what they're doing. Like for you and me, the minute you just slice through the skin, you're going to stop what you're doing. And so they have a lot of deformities, again, because they can't feel what the rest of us feel. So we'll be doing a little test with nociceptors today during class too, just so that I can watch you all have some pain. No, I was joking. Did I say that out loud? Sorry. I was just thinking it. No, sorry. Anyway. All right, so we're going to talk for a little bit about a group of cells that are found both in the central and the peripheral nervous system. And these cells are what we would call helper cells. They're there to help our neurons. As a matter of fact, in the central nervous system, we have more of these helper cells than we even have neurons. These helper cells are called neuroglial cells, okay? Satellite, 2 L's or 1? Satellite? Two L's. Okay, I got it right. Good. Okay, give me 1 second. Let me erase this. Type of neuroglial cell we're going to talk about is something called an astrocyte. Astrocytes have 2 very important jobs. Job number 1. It maintains the blood-brain barrier. Job number 2, it forms and holds synapses. Okay, so let's talk first about the blood-brain barrier. So in this picture here, this is an astrocyte. And in this picture here, this is an astrocyte. Okay? Now, astrocytes are always found right by blood vessels, which is what you see here in this red. These are blood vessels. So you can see it very well in the cartoon. This is also a blood vessel. And the astrocytes have these little legs, these little projections that come off of them. And the astrocytes in the brain will grab a hold of the nearest blood vessels and also grab a hold of the neuron. So in yellow, this is to represent the neuron. Now notice in the blood vessel, when the astrocytes grab a hold of it, they wrap around this blood vessel so much that you can't even see the blood vessel. Not the same thing thing with the neuron. They're just kind of sort of holding on to it. They don't totally wrap around it. The reason that these astrocytes are wrapping around this blood vessel completely is because they are forming a barrier, they're forming a blockade, not allowing certain chemicals to leave that bloodstream and get into the brain and the spinal cord. There are certain things we don't want to actually jump out of the blood and into our central nervous system. And the astrocytes have the ability to prevent that. The other thing that the astrocytes also are doing here is they're able to basically suck out, remove certain nutrients from the blood and then deliver those to the neuron. So the astrocytes are not only blocking particular chemicals from getting out of the bloodstream, but they're also taking other chemicals from the bloodstream, which would be nutrients for our neurons, to help keep those neurons alive. It's almost kind of like they're nursing the neurons. So they're taking chemicals from the blood and feeding our neurons. So that blood-brain barrier is actually the astrocytes blocking chemicals from leaving the bloodstream, and then astrocytes many times have been called nurse cells. They're feeding our neurons. All right, let's get started again. We are going to continue talking about the neuroglial cell. And if you can't figure out what this is, it's not a squid and it's not a carrot. It's the Dr. Harvey rendition of a neuron, okay? So I'll label it a little bit more later, but just get used to it now. So neuroglial cells. If we go back to the fetal stage of development, when we're first starting to develop the brain and the spinal cord, you have a lot of different neurons that are being produced. And those neurons have to be able to talk to each other. And so what the neuroglial cell does with all its little arms, what we call its podocytes, it will grab a hold of neuron number 1 and grab a hold of neuron number 2 and bring them together, kind of plug them in so that they can have a discussion, so that they can talk to each other. So if we say that this is neuron number 1 and this is neuron number 2, the information flows in this direction from neuron number 1 across to neuron number 2. Now, where these two neurons meet, this region here, this is what we call the synapse. So when those astrocytes bring these two neurons together to talk, they're talking to each other at the synapse. The small area between neuron 1 and neuron 2, this open space here, this is what we would call the synaptic cleft. Okay, now on the board it looks huge, but in our body, when these two neurons come together at this synapse, that synaptic cleft is very, very tiny. It's really small. As a matter of fact, it's been measured from the end of neuron 1 to the beginning of neuron 2, it is only 10 angstroms. So remember how tiny an angstrom is? Okay? Very, very small. Which means basically that these two neurons are almost touching each other. They are so close to each other. Okay? That synaptic cleft is not very big whatsoever. Now also, just so that you know, since this first neuron comes before the synapse, we call this the presynaptic neuron. And because the second neuron comes after the synapse, we're going to refer to this as the postsynaptic neuron. Okay? So then astrocytes continue to do this throughout the brain and the spinal cord. So they're going to get neuron number 68 and neuron number 572 and bring them together so that they can talk to each other and form all these synapses. Now in our brain, astrocytes astrocytes form, and our spinal cord, astrocytes form two types of synapses. One type of synapse is what we would call plastic. And another type of synapse is what we would call cemented. Okay? So let me tell you what the difference between a plastic synapse is and a cemented synapse, because one of the terms you're going to use in the medical field is you're going to try to say, hey, we need to keep as much plasticity in our patient's brain as possible, because you want more plastic neurons than cemented. So here's plasticity. Anytime I learn something, doesn't matter what I'm learning. So maybe I'm going to learn an instrument. Okay, maybe I'm going to learn to play the violin. All right? As I learn to play this instrument, what's happening in my brain is that my astrocytes are actually moving neurons around, so that different neurons can start talking to each other that may never have talked to each other before. And the more I practice at playing this violin, the easier it is for my astrocytes to move these neurons into the appropriate place so that I can improve. And the more I improve, the more astrocytes can be moving neurons around so that I can play better and better. If an astrocyte has the ability to move neurons around in different places, those neurons that are being moved, those are plastic neurons, and we form plastic synapses. But plastic synapses may not stay where you put them. So I'm practicing this violin, and maybe I get really, really good at playing the violin. Maybe I even play like in an orchestra or something. And then I'm just sick of it. I don't want to play this violin anymore. I put it in a case, I put it away, I don't have anything to do with it for the next 20 years. And then I'm regretting it, and I get the violin back out again. Am I going to be able to play as good as I did 20 years ago?
No.
Now, I might remember things. I might still be able to play somewhat because I did a lot of practicing, but I'm not going to be as good. The reason is because over those 20 years, I was doing different things. And the astrocytes took some of those neurons that were there all about my music playing and moved them to different places because I started doing different things. And the astrocytes started moving neurons. If I want to get really good again, I've got to bring neurons back, but now here's the problem. Sometimes when astrocytes bring two neurons together, they will stay there forever. And they will become what we call cemented. So I'll give you another, for instance. Riding a bike. Once you learn how to ride a bike, you will never forget. So if you haven't ridden a bike for, let's say, 30 years, and then you get on a bicycle to go ride it, you can. You will be able to. You might look a little klutzy, but you will be able to still ride that bike. That's because once you learned how to ride that bike, those astrocytes, while you were practicing, were bringing certain neurons together, forming certain synapses until they formed enough that your brain was able to go, okay, I got this balance thing, I understand what's going on, let's ride. No more training wheels. But for whatever reason, and nobody really knows how the brain decides which synapses are plastic and can be moved, and which synapses are cemented and can never be moved. Riding a bike is all about cemented synapses. And so once you learn how to ride it, those synapses are always there, and they will never be moved, and you will know how to ride a bike when you're 90 years old. Now, you may not be able to go, oh, look, Mom, no hands, okay, but you'll still be able to get on and ride because those synapses are cemented. Now, some of you may have experienced this when you came to school. So let's say that, you graduated high school and then you waited for several years to come back to college. In your first semester, you probably thought, oh, I got this. I did pretty good in high school. And then you realize, I don't really got this, okay? Things have happened, and I forget how to study, and I forget how to memorize, and I forget all this stuff. But maybe by the second semester, it was starting to get better. And by the third semester, you're like, oh yeah, I got this. There's no problem. Well, what was happening all that time is as you were practicing, studying, and doing the things you needed to do for school, astrocytes were moving synapses again. And in our elderly patients, one of the problems is if you're not constantly utilizing the brain, it becomes harder and harder for astrocytes to move these synapses. It becomes more difficult. And it leads much more likely to things like dementia. So the more you have people using their brain, I mean, even just playing games, doing different things, reading fun books, doesn't matter. Something besides sitting around watching television all the time, okay? This will help to keep the brain more plastic, more flexible, so that it's easier for them to learn and less likely that they're going to have dementia problems. Any questions?
About how long does it take for you to start losing that connection to like if you were like say you're really good at sport and you haven't played like in 3 years? Is it about gone at that time, or is there like
Well, it's not necessarily completely gone. I mean, if you've been playing the sport for, let's say, 10, 15 years, and you let off for 3 years, no, it's not going to be completely gone. But if you've only been playing the sport for 2 years, and then you didn't play for 3, yeah, it's probably gone.
No.
Yeah.
Let's say, for example, because if a person like study biology for 4 years, and then after 4 years he's done, and then after 10 years, maybe he forgot about it. But how about the memories? Keep on, you know, like...
You're not going to know anything.
No, I mean, your memories when you were a kid, it's stuck in our, like...
Oh, I
You
see what you're
remember
saying.
that.
I see what you're saying. So you're talking about childhood memories versus I go to college and then a couple years
Study
later I don't
for-
remember what I studied?
Mhm.
Okay. Well, there's a couple of things about that. First of all, childhood memories, for the most part, if we're talking a healthy childhood, there's not a lot of stress. And because you don't have a lot of stress, your brain remembers things easily. In college, you're under tremendous amounts of stress. Your brain remembers nothing. The more stress you are under, the more difficult it is to remember things.
That's what happens when we work.
Yes, exactly, exactly. It's unfortunate, but it's true.
Oh my gosh.
All right, the next type of cell is called a microglial cell. Microglial cells are phagocytes. So their job is to move around the central nervous system and eat up things that don't belong there. Dead tissue, let's say maybe you have a patient who had a stroke. These microglial cells are going to get rid of all that dead tissue that's there and help to clean up the area. Maybe there's a virus that somehow gets past the blood-brain barrier. The job of the microglial cell is to try to kill that virus. Okay? Its job is to make sure that the central nervous system keeps as clean as possible. One of the problems, though, microglial cells may also be carriers of certain viruses into the central nervous system. Because a microglial cell can actually leave the central nervous system. It can get into the bloodstream, pass through that blood-brain barrier, and get into the peripheral nervous system and cruise around there too. So let's say, for instance, you have a patient who has HIV, and this virus is in their system. That virus likes to get into phagocytes. So it'll get into the microglial cell, which then crawls back through the blood-brain barrier into our central nervous system and drops off the virus. This is why some, not all, but some of your AIDS patients may end up with dementia because the microglial cell took the virus into the brain. The next type of neuroglial cell is called an ependymal cell. Ependymal cells are only found in the ventricles of the brain and the spinal cord. So if you remember, we've got the lateral ventricles, third ventricle, fourth ventricle, and then the middle of the spinal cord is hollow. So all these ependymal cells are lining the walls of all of these ventricles. And if you look here on the overhead, you can see the cilia on the ependymal cells. So the ventricles produce cerebrospinal fluid. And the job of the ependymal cells with its cilia is to move this cerebrospinal fluid. It has to move around the brain, around the spinal cord. This fluid is helping to keep the brain and spinal cord moist, but it also has a lot of nutrients in it. And so the ependymal cells are constantly moving that cilia all in one direction, and that CSF is flowing around the brain and spinal cord in one direction, and it's providing nutrients to the central nervous system. Okay, now sometimes things can get into the cerebrospinal fluid like viruses, bacteria, something like meningitis, and really gunk it up, get it really thick, and it doesn't flow well. The cilia can't beat hard enough to get it moving, and this can cause some serious brain damage, even kill your patient. Oligodendrocytes. Oligodendrocytes produce myelin. And they make a lot of myelin. They make enough myelin to cover at least 60 neurons apiece. That's a lot. So in this overhead, this little cell here that kind of sort of looks like an octopus, this is an oligodendrocyte. And notice the bundles of cells underneath this oligodendrocyte. These are neurons. So these little oligodendrocytes can move, and they kind of crawl up and down these bundles of neurons, and they're constantly looking to see if any of the myelin has, kind of torn off or been scraped away, and they put more myelin back on and repair it. So they're constantly making sure that insulation is nice and thick over top of all of these neurons. This is, in yellow, also an oligodendrocyte, and in red, these are all the neurons that are around it that it is producing myelin for and keeping it insulated. Okay, so in the peripheral nervous system, we have two types of neuroglial cells. The first type we're going to talk about is actually the satellite cell. And this is going to be really simple because they're still not sure what the heck the satellite cell does. The next cell is the Schwann cell. And like the oligodendrocyte, the Schwann cell produces myelin. But it doesn't produce very much myelin. Okay? So notice in this overhead right here, this is myelin right here from a Schwann cell. And then here's a little bit more myelin from a Schwann cell. So all these little white pieces here that's on this axon, this is all myelin from a Schwann cell. That's not very much myelin. What the Schwann cell does is it grabs a hold of the neuron and then starts making myelin that wraps around and around and around and around, so that you kind of have like this sort of, you ever seen a jelly roll? That kind of donut that wraps around and around. Anyway, the myelin goes round, 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 round this Schwann cell, but there's only a small amount of myelin. So you have to have multiple Schwann cells to cover this neuron. Not like the oligodendrocyte that has enough myelin, it can cover 60 neurons. The Schwann cell can't even cover